Moving on to the nucleic acids. So these are also polymers. So these are going to be made up of nucleotides. Nucleotides are going to consist of a nitrogenous base, 5-carbon sugar, and a phosphate group. So remember, for this 5-carbon sugar, we talked about that OSE ending. Okay, so this is going to help us to remember that these are sugars, and the deoxyribose would be the 5-carbon sugar of DNA, and the ribose would be the 5-carbon sugar of RNA. Now, these polymers of nucleotides are going to be linked by phosphodiester bonds. Phosphodiester linkage is how we're going to be building these. And we will get into more detail on these as we move through the year. Okay, this phosphodiester linkage is going to form the backbone, the sugar phosphate backbone of our nucleic acids. So the two types of nucleic acids that we need to be familiar with at this point are RNA and DNA. So RNA is going to provide the codes for the amino acids in the proteins. So we start with DNA. DNA codes for RNA. A copy of RNA is moved out to the ribosomes and the cytoplasm, and then that is then going to be translated into a chain of amino acids, which will ultimately lead to our proteins. And we'll talk more about protein structure coming up. Our DNA is going to be a double helix formed by complementary base pairing. Okay, so the bases, remember, are going to be our purines and our pyrimidines. You're probably more familiar with the A, T, C, and G. So adenine and guanine are going to be purines. These are actually double ring structures. And then our pyrimidines are cytosine and thymine of DNA, but then uracil would be found in RNA. All right, so let's go ahead and move on to proteins. We're going to be here for a little while. Lots and lots of information. So first of all, proteins are going to be polymers. They are polymers of amino acids, which means that amino acids are monomers. So the monomers of proteins are called amino acids. Okay, and these amino acids are going to have a specific structure. Okay, we're going to have an alpha carbon, so it's going to be a central carbon, and remember that carbon can bond to 4 different atoms. Okay, well, one of the carbon electrons is going to be shared with something called an amino group, which is going to be an NH2 group. So you can remember the N for amino. On the opposite side of that amino acid is going to be a carboxyl group. This is going to be the same carboxyl group that we're going to find on the end of our fatty acid chains. Okay, on one side we'll find hydrogen, just a hydrogen atom. And on the opposite side of that, we'll find what's called the R group. This is the reactive group. This is the group that gives the amino acid its specific reactivity. Okay, how does it function? How does it interact with other things? That's all going to be determined by these R groups. So this is going to contribute to something called tertiary structure. It's also going to be involved when we talk about quaternary structure as well. So these R groups are the reactive groups. And there are 20 different amino acids, and they differ in this group here, this R group here. Everything here, these 4 items, the central carbon, the amino group, carboxyl group, and hydrogen, are identical. They only vary in the R group. The polymer of amino acids is going to make the primary structure. So when we talked about nucleic acids, we said DNA codes for RNA, RNA goes to the ribosomes are then going to code for our proteins. Well, what's happening there is the primary structure. It's just a simple chain of amino acids. These amino acids are going to be joined by peptide bonds. Okay? Now, the peptide bond is going to form between a hydrogen from an amino group of one amino acid and the OH group of a carboxyl group from another amino acid. And proteins, their function is going to depend on the shape. Okay, the shape is going to be dependent upon the sequence. So our primary structure is the chain of amino acids. When we start to look at secondary structure, okay, secondary structure is going to be determined by the interaction between the different R groups. Okay, so the 3-dimensional shape results from primary structure, which is going to then determine the secondary structure, and our secondary structure is going to determine our tertiary and quaternary structures. So in a secondary structure, you're going to either get an alpha helix, so think a single side of a DNA molecule, that's a helix. Okay, that's spiral helix. Or beta pleated sheets, which basically take a piece of paper, and if you keep folding it back on itself, that would be what a beta pleated sheet would kind of look like. Okay, this secondary structure is going to be created because of the hydrogen bonds within that polypeptide backbone. Okay, remember hydrogen bonds, that weak charge between the positive and negatives. Okay, it's also going to determine our tertiary structure, okay, which is going to be created by something called van der Waals forces. Remember that weak interaction between these charges. Ionic bonds, remember they are exchanging electrons. Okay, so an ionic would be between a positive amino acid and a negative amino acid. Disulfide bridges, which you're going to see when you're talking about cysteine, you'll see that it has a sulfur atom in there, and that's going to form a disulfide bridge. And then the hydrophobic interactions. Remember, you might have some that are hydrophobic or hydrophilic. Okay, so they are going to either hydrophobic will try to get inside the protein, okay, and hydrophilic will stay towards the outside. And then your hydrogen bonds as well for tertiary structure. So now tertiary structure is then going to help determine the quaternary structure. Tertiary structure for a single strand of amino acids is going to be the most complex that it will look like. If you go to quaternary structure, now what you're doing is you're adding in another protein chain, and they are reacting together. A good example of something that has quaternary structure would be a hemoglobin molecule. It's actually made of 4 different protein strands. Okay? So the function, talking about tertiary or quaternary structure of proteins. 1, so they can be structural. Think fibers, your spindle fibers, your muscle fibers, okay, actin filaments. Think myosin filaments. Think cytoskeleton. They can also be transport. So I already mentioned hemoglobin. And we also have membrane proteins. So these are going to allow molecules to move in and out of cells. Movement. I already mentioned the actin and myosin fibers in muscles. Hormones like insulin. We'll talk about those a little bit later when we get into cell communication. Defense, antibodies. And then they speed up chemical reactions. And this is probably one of the most important functions that they do in speeding up chemical reactions. They are acting as catalysts. And in this case, we're talking specifically about enzymes, which are catalysts found in living systems. So here was your review of organic molecules, and I will see you guys in class.